You're listening to the Future Tech Health Podcast with Richard Jacobs. Until I reached age 40, I never realized the obvious, that we all have medical issues, or we at least have a family member or close relation that has, or will have them in the future. Medicine and biological systems are the final frontier. Until we've conquered death, figured out how life began, cured cancer, and understood our purpose in the universe, there's a heck of a lot to talk about when it comes to our health. Future Tech Health means I'll be covering futuristic topics that are actually already in clinical trials, or even starting to appear on shelves or by prescription, or available for your own use. We dive deep into stem cells, CRISPR-Cas9, the science of sleep, epigenetics, medical testing, cancer, ketogenic diets, stem cells, aging, regenerative medicine, and more. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a serious medical problem. Remember, however, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you enjoyed the podcast, please listen, subscribe, like, and share it with friends. Thank you. Hello, this is Richard Jacobs with the Future Tech and Future Tech Health Podcast. I have Patient-derived. He's the CEO and shareholder of CELLphenomics. They make patient-derived organoids, 3D organoid models, which we'll get into in detail. So, Christian, thanks for coming. How are you doing?
Thanks for the invitation. I'm fine, and I'm happy to discuss what we are doing with you and your audience.
Oh, good. Well, tell me what organoids are being made by a large number of groups. What's the focus of your group?
So the origin of what we are doing is from a pan-European project called the IMI Initiative OncoTrack, where we started generating organoids from colorectal cancer. And it was a partnership with FPIA, meaning that also pharma partners were part in that consortium. And they were asking for, could you do drug screening for us on the organoids? And together with colleagues and now friends from Eli and Libby, we developed a pipeline for high throughput drug screening on a 384-well format. And yeah, what we did then is we were screening substances, single substances and combinations. We did it quite successfully. And so we started our own company in 2014. And then I moved from academia totally to the new company in 2016. And yeah, just this year we founded a second company which focused on bringing this technology directly to the patients and oncologists.
So you take a patient's tumor cells and you culture them into making a tumor outside of their body, in like a lab environment so you can study it?
Yes, that is in principle correct. So we receive either from surgical specimen or biopsies, pieces of the tumor. Ideally, we also receive a bit of patient blood, like 10 to 20 mil. So we can do co-culture models with autologous immune system or immune cells from the patients. And yeah, what we're doing is we cultivate those cells, and we have established protocols that allow us to start the screening process within like 3 weeks from most of the solid tumors. And then we do the drug screening of substances that oncologists suggest that could be benefiting the patient.
So you deal with people that have colorectal cancer. Do you take cells from just one of their tumors? I mean, do these people tend to have multiple tumors or just one?
No. So what we ideally do if the tumor, unfortunately for the patient, is big enough and we can receive samples from multiple regions, we can also investigate the intra-tumor heterogeneity, meaning the different kind of clones that are laying in the same tumor. And we have very interesting results generated with that. For example, we found in one patient where we had 5 regions, that within these 5 regions, there is an up to 30-fold difference in the response to standard of care drugs, and that only a combination of drugs will lead to a sufficient effect on the tumor. And that was for me really a eureka moment, because until that point of time, I always thought, okay, we're in the 21st century. Why is the first-line treatment of colorectal cancer still a classical chemotherapy and not a targeted therapy? And now I know it's because of the heterogeneity. You try to remove the amount of clones, reduce them to a smaller population, and then with targeted treatments, you may have a better chance to hit those tumors.
Well, you're looking at different regions of one tumor, but what about if someone has more than one tumor in their colon? What if they have 3 or 5 or 20 of them? What about sampling different tumors themselves to see if the heterogeneity goes to a new level?
That is almost the trivial thing, doing the different tumors in the same organ or spread throughout various organs. And what we do is we cultivate those separately wherever we can get hold of the tissue and try to compare them and do predictions. Interestingly, it also makes a difference in the drug screening process if you have only a single population and do a screening, or if you co-culture different populations from within the same tumor and do drug screening then. So it seems that there is also a paracrine signaling between different cell types, either from the same tumor or multiple tumors within the same patient. And that is something we are currently investigating and learning a lot from.
Well, if there's multiple tumors, can you identify a lineage amongst them? What I mean is, let's say someone's colon has, 10 tumors. You sample them all. Can you tell which one arose first, and do the other ones arise from the first one, or do they seem to arise independently?
That is something that is very different from patient to patient. So there are some patients where you have, I would say, like a Cambric explosion of mutations very early, and then you have very many populations sitting side by side, and then it's very difficult to do a reconstruction. But then you see in other tumors or in other patients that the tumors harbor always the same driver mutations, and then an additional set of passenger mutations which add up to it. And there you can do tumor reconstruction. So we have published earlier this year for one patient such a thing where we did a tumor evolution pattern together with some bioinformaticians.
Well, what have you noticed about how one particular tumor evolves over time? I mean, supposedly a given tumor starts with one cell that just keeps dividing and then morphing and mutating. Is that the theory? And do you observe that?
Yes, of course, there must be the one tumor cell that started all the process. But it is not said that during the process of tumor development and at the stage where you identify and diagnose the tumor, that you really find this cell of origin. It could be that this is a clone that does not survive the entire process and that dies off early. So we cannot really say if what we see when a tumor is clinically diagnosed is really the setup, or the cells with the fused mutation is really the tumor founding cell. It could be that this is just coming up later, because usually when you can identify and diagnose tumor, you have 10 to the power of 8 to 10 to the power of 9 cells already in the tumor, making it very difficult to determine, is this really the progeny of the tumor founding cell?
So it's okay. So you have a lot of cells, I understand, but when you look at the tumor, what if some of the cells only have 5 mutations, and then some of them have 10, and some of them have 50? Perhaps that would mean that the ones that have 50 were the ones that used to have 20 and used to have 5, and there's a lineage. There's there's descendants of the original tumor cell. Has anyone been able to tell such a thing, or is anyone looking even for such a thing?
Yes, there are multiple studies, and I think who's really leading the field is Marco Gerlinger in the UK, who also published the first real genomic paper on intratumor heterogeneity in 2012 in New England Journal of Medicine. He I think, is the one person that you should go to if you're interested really into the development of the intratumor heterogeneity. Because at that point where you have mutations in the DNA damage repair system, the accumulation of mutations gets really weird and wild. So that is the point where we cannot, or we are not interested in doing the tumor reconstruction on an evolutionary basis, but try to map it back to the drug response, because this is how we hope to be able to help the patient.
Yeah, no, I know you're trying to evaluate drugs. Mm-hmm. So, we'll turn the questions back there. How heterogeneous have you seen a given tumor to be? Are there, how many, you spoke in the beginning, there could be like regions of heterogeneity, but, within a tumor, how many different types of cells are there that you've seen?
So that is also very dependent on the stability of the genome. So if you have a microsatellite unstable tumor, you would expect much more heterogeneity than on a microsatellite stable tumor. And same is true about the origin of the tumor. So if the tumor is, for example, caused by an oncogenic agent, then one has specific mutation patterns that you always find throughout the genome, but there can be also virally induced tumors which have a more difficult pattern to decipher. And we must not forget that all the things we are just talking about mostly apply to carcinomas. And there are also soft tissue tumors, sarcomas, which are on the genomic level totally different. You find either just a few mutations, which are usually fusion genes, or the genome of such a patient or such a patient's tumor looks like if you have aimed at it with a shotgun, and a variety of mutations which are not seen commonly, for example, in xenomas. So that is also dependent on the entity that you're looking at.
Yeah, the reason I ask about the evolution of the mutations is that if you're able to identify a core set of mutations that all the tumor cells share, that seems like the most targetable thing for a cocktail of drug therapy, not the individual mutations that they all have. If there's 1,000 different variations, how would you ever target a tumor? I would think they would have to have a core set of mutations you can go after. So in order to find that, you would have to look at, the lineage and the progression of a tumor to see what's preserved.
Yes, either that, or you really have to delayer the tumor mutation by mutation and say, okay, let's say 90% of the tumor volume of the cells contain a certain RAS mutation or a PI3 kinase mutation or whatever driver mutation you imagine. And if you kill off that, this already reduces the tumor volume by large. So it doesn't make too much sense if, for example, we just had a tumor where we found a mutation in one of our cell cultures at 100%. When going back to the original patient tumor, we had to sequence with a 2000x coverage, and only then we were able to see this specific mutation. So I would say, in regards of treating the patient, first aim at the most prominent mutations, or start, as it is currently done clinically, with a classical chemotherapy that hits all rapidly dividing cells equally.
Also too, the distribution of the mutations. What if the tumor, let's say it's like an orange, it has a rind of certain mutations on the outer part, and then in the inner part it has different ones? Would it make sense in a case like that to target the outer stuff first with a drug, and then after a short period of time then start targeting the inner stuff? Perhaps the order in which a drug is given would be critical if it had morphology.
Yes, that could definitely be critical, the order in which you hit the cells. But it is already known since at least 5 to 10 years that you have either migrating cells or proliferating cells. So I would say it is dependent on the situation the patient is in. If you want first to hit those cells that generate the neoplasm, the new growth of tissue, or if you try to hit those cells that tend to migrate and give rise to metastases. For example, there are the Mullerian mixed tumors, and these tumors have just micro primary tumors, which in about 90% of the patients are not found in the body, but they give quick rise to growing metastases. So at these tumor types, it would be crucial to stop those cells from migrating into the body and starting metastases.
Yeah, I guess, yeah, the metastases would be far worse.
Right.
Which, in your observation, which is more targetable? Which cells are more susceptible to, to the effect of drugs? The metastasizing migrating cells, the original tumor cells, the proliferating ones?
So all cells that do have a rather quick metabolism or divide quickly, these are the cells that are better to target. Of course, we have also to observe their effects like ABC transporters or PGP, P-glycoprotein, which are active pumps that help the tumor to fight against any kind of chemotherapy by actively pumping the substance out of the tumor cell. So that is also something we have to keep in mind, to block these kind of mechanisms in order to make the treatment really work.
Yeah. You mentioned signaling amongst the various tumors. What are you looking at in that regard? Have you seen signaling amongst the organoids that you're culturing?
Yes, so together with colleagues from the NMI, which is a private research institute in southern Germany, we have developed an approach to do targeted oncoproteomics, meaning it's a Western blot-based method, and we can interrogate up to 200 antibodies, the proteins and the phosphoproteins, meaning the post-translational modifications. This gives us a very good impression about the signaling pathways that are used by the cell. And as we can do it in vitro, we can do time resolution experiments, seeing the immediate early effects and seeing the later effects. So we can also get an understanding of how cells try to circumvent the treatment. And so what I see as a big point in the future that will enable us to fight cancer better than we are able to do it today is proteomics, specifically targeted oncoproteomics, with only a limited set where we have little amounts of tissue that we need, but get a direct answer to which pathways were activated, which pathways are used to circumvent the treatment, and how to do a double blockage by combinatorial treatment.
What about exosomes that would be given off by the tumors? I've heard that tumors do give those off, and especially if they're exposed to certain drugs, they tend to give off like tons of exosomes. Are you looking at those?
Yeah, I'm happy that you're asking for that, because we just this month started a project together with the University Hospital in Göttingen on exploring the exosomes. What we already know from preparatory experiments is that we can isolate the exosomes that are secreted into the cell culture medium and use this to decipher the exosome load. I know that there were like 2 or 3 papers mentioning that the exosome load gives you an idea of where the next site of metastasis will be. And this is something where we want to compare if the in vitro signals that we receive in our organoids are comparable to those exosomes that can be identified in the patient.
And then another thing I've heard of, which makes it even more complicated, is that supposedly each tumor has its associated microbial fraction, or its own microbiome, I guess you could say. Mm-hmm. I don't know how you recreate that when you're making organoids, but how critical a part do you think that is?
Oh, that is really a good question, which I honestly cannot answer at that moment. It's just educated guessing what I can do. So we all know that the microbiome seems to be stronger or more influencing than we thought like 5 years ago. But I haven't heard of any convincing experiment in vitro reconstructing the microbiome for organoids or for tumors in regards of how this could link and translate to patient treatment.
Well, how much of the behavior that you're seeing appears to be unexplainable so far with the current organoids that you're making and the models you're using? Is there a vast amount of behavior and things that just are mysterious, and perhaps it's because the model needs to be, improved somehow, maybe by including the microbial fraction or considering other types of elements? I mean, I know it's a big guess, but overall, what do you see?
So in regards of looking into the glass ball and looking into the future, I would say the next level of evolution of in vitro models, organoid models, will be co-culture models with immune cells, which are currently undergoing in a certain amount of labs. Then we are currently exploring together with people from the University in California, doing angiogenesis co-culture models with organoids. And what I think in regards of being able to help the patient will be a great step forward if you combine iPS technologies and organoid technologies to mimic the metabolism of a patient by generating iPS liver cells, which are then patient-specific, and see how a certain drug or combination of drug is metabolized by that specific patient. And then you can, I hope for the patients, much better distinguish which amounts of drug would you really need, because the current standard for most of the drugs is body weight and body surface, which is, I would say, not exactly rocket science.
In terms of the efficacy of what you've found already, I mean, how much better are you able to identify? You said you have a high throughput model now for testing drugs on these organoids. So when you say high throughput, how many drugs are you able to test and how many combinations?
So when talking about the 384-well plate, we can test up to 12 substances easily in statistically relevant multiplicates. In terms of drug combinations, we usually do a 6 to 6 matrix of 2 drugs and identify how those interact with each other. This gives us enough resolution to observe not only additive effects, but also synergistic effects, where we can then see that small changes in the concentrations really lead to a jumping high effect on the tumor, which then means that we would be able to reduce the overall concentration of chemotherapy, meaning in the best case scenario, we can smoothen a little bit the side effects, but still have a very strong effect on the tumor. Let me give you one example. We had a tumor, colorectal cancer again, with a RAS mutation, and of course, a colorectal cancer patient with a RAS mutation would not be prescribed cetuximab, and we tested it in vitro, and cetuximab alone didn't work. The same was true for regorafenib, but the combination of cetuximab and regorafenib for this patient was really, really the best thing that we have tested in the patient, and we have tested like 12 single substances and a combination thereof. And so we have tested this then in an animal model with the same patient tumor, so a PDX model of the same tumor, and we could there really clarify that this combination also in vivo was the most effective. And this brings us to a brand new level of exploring how patients can be treated in the future.
Have you seen, with a given chemo regimen, just by changing the various levels of the drugs involved, does that create a dramatically different effect? Like what are the big levers that seem to affect tumors very strongly based on your models and the insights you're getting?
The most effective thing that we see is double blockage of the same pathway. For example, if you combine a MEK inhibition with an ERK inhibition, or something at the receptor level with something more substantially downstream of the same pathway. That is very, very effective. And also, if you look at intersection pathways, for example, where mTOR meets PI3 kinase, if you're blocking at those nodes, then you can be very, very effective in the drug treatment.
Okay. Anything you've seen that really surprises you in its efficacy, or do you have any ideas or pathways to go down? I don't know if you can disclose them or not, but What are some areas that you really want to explore you think would be very impactful?
What was very surprising to us was that in this colorectal cancer model, cetuximab plus regorafenib were working together like a charm and had high synergistic effects because each drug alone didn't work at all, and it wouldn't, from the clinical use, wouldn't be considered to even administer cetuximab to this patient. And I think these are the moments where you can really appreciate the organoids, because you have the ability to test things on a broad basis without killing or harming the patient. And this is, I think, the big fascination and the big issue that we are happy working with.
And how fast can this be done? Someone has active cancer. I guess during a surgery, during a resection, that's when you're getting your material. And how fast can you do a turnaround to tell them, all right, try this, try that?
So our absolute fastest test was with a patient where with a breast cancer, where we had 9 days from surgery to we had tested the substances that the oncologist would have considered to administer to the woman. So 9 days was the best. In average, we need about 4 weeks.
Well, okay. And then you'll speak to their team that's treating them and say, all right, this is the particular chemo regimen you need to give them. And you tailor it, certain levels of drugs, certain drugs being there or not there, length of treatment, that kind of thing. Or do you leave that tailoring to the doctors on hand? Like, how do you advise on what to do based on what you find?
So what we do when working together with university hospitals, we participate in the molecular tumor pathology conference. So if there's a molecular tumor board, we discuss with the oncologists and the sequencing people the pros and cons of different regimens. And we leave entirely to the oncologist how he or she treats the patient, because at the end of the day, this is the person responsible for the benefit of the patient, for the life of the patient, and for the well-being. So this person is in the driver's seat no matter what. No matter how sure we are, the oncologist needs to justify whatever he or she does to the patient and the family, and therefore he or she is in the driver's seat.
So are you at the point where you've now advised and a different regimen has been given to actual live patients, or are you not there yet?
No, we are already there. And when we do it like a co-clinical trial, so we do suggestions, the oncologist treats the patient, and if the oncologist decides to treat the patient differently from what we have suggested, we also test what the actual patient is given, and then we do co-trial and see, does the patient benefit? Do we predict the response in vitro, what happens in the patient? And this is currently ongoing with a couple of university hospitals here in Europe.
Have you gotten any results yet that you can look at? Are you seeing any difference in results or no?
Yes, we see already results which are very encouraging and which helped us to extend those trials. But as this data is not published yet, I would rather not comment to this in more detail. But there will soon be something published in this direction.
All right, but it is encouraging so far.
Yes, it is, definitely.
Oh, that's great.
Let me put it like this. I would never allow a chemo if I had a tumor which was not tested before.
Yeah, it makes sense, because I guess it's just a bludgeon that may not work at all, depending on what the cell types are in that person.
There was, I think it was by the end of 2018, a publication by Vlado Janis and Nicola Valeri in the UK, where they tested, I think it was 115 colorectal cancer patients, did a co-clinical trial, and were correctly predicting not working chemotherapies or targeted therapies with 100% negative prediction value. And I think the positive prediction value published in that paper was at 88%. So comparing this with today's standard of care results, 88% and 100% great numbers. And this is very encouraging.
Yeah. How expensive is it to do this process? Like for a person, how expensive do you think it will be to have this done? When will it be? Is it very affordable, or is it still very expensive, or where is it at economically right now?
Well, that's the question of which price tag do you put on a human life if you consider it expensive or affordable. But we are currently at a price point of about €5,000 for testing up to, I think, 5 substances. And if this is a decision of life or death, €5,000 seem to be quite a fair number.
Yeah. How many substances do you guess would be needed to test in order to really identify what a person would need or not?
I think this may be significantly higher today because we are just at the beginning of the learning curve, but I would expect it to decrease over time. So today, what we are doing as our primary effort is there's, of course, always a limited set of substances which the oncologists consider themselves to use. And this is the primary set we are testing. If the cells are growing well, and if this is a case we are very interested in, we just over time test more and more substances and combinations for the same patient without taking into consideration that this is clinically relevant. This is just for our learning curve. But yeah, that is the research we are doing in our company.
Okay, so an oncologist may only have 7 substances total that they can use. You can at least test those different combinations, strengths, etc. Okay.
Yeah, I can give you the example of what I told you before about the cetuximab and regorafenib. Even when we, on a scientific level, were publishing the data earlier this year, one of the reviewers was, why did you test cetuximab at all? Because cetuximab would not be administered clinically to that patient. And the simple and trivial answer to that question is because we can. And we can do it in vitro. It doesn't harm the patient. It doesn't take more time because we can do it all in parallel. And whatever educated guess will bring us closer to healing a specific cancer is something I'm happily doing. And in theory, we could also in parallel just look at the clinicaltrials.gov website, and from there also test already for specific patients clinical trials that are currently open, and then suggest to the oncologist to administer this patient into the specific trial because we believe this could benefit the patient.
And is there a tumor database for different organs, like colorectal tumors? And are you contributing to such a database, because you're characterizing and culturing tumors from dozens, hundreds, and eventually maybe thousands of people? Is there such a thing that exists? And do you use that data and do you contribute to it?
So there are commercially available databases like Repositive.io, which offer databases for cancer models, which then can be used by biotech and pharma. Or there are public databases like the ICGC or the Cancer Genome Atlas, and these databases can contribute to our understanding. So what we are currently developing in-house is a database where we try to correlate the genomic level, the drug screening level, and the proteomic level, because there's, at this point, not a best practice approach to sync this kind of data set. And this is also something we are currently exploring, and we were granted some money from the Berlin Senate to explore these things.
Okay. Well, Christian, this has been very interesting. I hope you're okay with the range of questions. And for people that are interested to follow your work and to wait for the new papers to come out, where can they go? How can they find out what you're doing and keep tabs on when your new work is coming out?
So we have set up two websites. One is www.CELLphenomics.com, and the other is asc-oncology.eu. The first one is for the industry. The second one is for doctors and patients. And I'm happily answering all questions arising from patients or doctors via email or by telephone. So happy to communicate and get the message out there. And one last thing from my team, when they heard today that I'm giving an interview that will be a podcast, they said, could you send us some greetings, which I now happily do. And if you don't cut it out, so they know that I did the greeting thing.
No, we'll keep it in.
Thank you.
That's great. That's great. Thanks for coming. I appreciate it.
Thank you very much.
You're listening to the Future Tech Health Podcast with Richard Jacobs. Until I reached age 40, I never realized the obvious, that we all have medical issues, or we at least have a family member or close relation that has, or will have them in the future. Medicine and biological systems are the final frontier. Until we've conquered death, figured out how life began, cured cancer, and understood our purpose in the universe, there's a heck of a lot to talk about when it comes to our health. Future Tech Health means I'll be covering futuristic topics that are actually already in clinical trials, or even starting to appear on shelves or by prescription, or available for your own use. We dive deep into stem cells, CRISPR-Cas9, the science of sleep, epigenetics, medical testing, cancer, ketogenic diets, stem cells, aging, regenerative medicine, and more. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a serious medical problem. Remember, however, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you enjoyed the podcast, please listen, subscribe, like, and share it with friends. Thank you.